In considering this question, it helps me to remember that life began on our planet more than 3.5 billion years ago. And for the first next 3 billion years, life existed as single-cell microorganisms, bacteria, archaea, and then single-cell eukaryotes. And they were so successful that the oxygen concentration atmospherically was increased rather dramatically and suddenly around 600 million years ago. And about that time, single-cell, multicellular organisms began to evolve. And within a relatively short period of time, it's thought about 10 million years, there was an explosive development of metazoan multicellular species with different body plans. Now, all of these creatures, as we all know, had innate ways, had immune systems, ways to defend themselves, some of them quite complex, even in bacteria and archaea, for defense against bacteriophages, plasmids, and we know a lot more about the toll-like receptors and other pattern recognition molecules from studies in flies, but only in our vertebrate lineage is there an adaptive immune system that features long-lived lymphocytes, each with its own specific receptor, able to recognize specific antigens and with the memory of that response. About 10 years ago, Jan Klein and I joined forces to try and go back and look for the origin or the roots of our adaptive immune system. And we knew to start with that all jawed vertebrates have a thymus where they generate T cells with BD and J recombination, B cells generated in hematopoietic tissues, MHC class 1 and 2 to present antigens to T cells. But there weren't any of the more basal, immediate progenitors or organisms because we know of them only through fossil remains. Apparently, most of them died out around 400 million years ago, except for these jawless vertebrates. There are two that are still extant, hagfish and lamprey. And our studies are mainly in the lampreys. And we found to our surprise that they have indeed an adaptive immune system that in many ways resembles ours, but the receptors that they use are totally different. They use leucine-rich repeats, which are used in many ways for body defense and many other purposes in all living organisms, and all the way back to plants where resistant genes involve use of the leucine-rich repeats instead of the Ig-based domains that we use. Now, this isn't the greatest model species to study. It's genetically intractable. We study them as outbred animals caught in the wild. They live relatively long, up to as old as 15 years, and much of their life is in a larval phase. The ones we study are predators, as about half of the 40 plus species of lampreys that are worldwide in distribution are, and they prey on fish as adults. They then, after about a year and a half of this predatory phase, come back into the streams that flow into the sea, and the male and female mate. Large numbers of eggs are fertilized, and life cycle begins again. These animals don't have these eel-like fish, don't have obvious lymphoid organs as we do, no obvious thymus, no spleen or lymph nodes. They produce blood cells as larvae in a typhlosaur, which runs the length of the intestine. And the strategy we began with was to get cells that were lymphocyte-like in light scatter capabilities and do a transcriptome analysis. And we were quite disappointed in not finding any of the cardinal genes for our adaptive immune system. The immune responses, though, in the form of very specific hemagglutinins had been reported many years earlier, and also allograft rejection had been seen. So we stimulated them with a cocktail of live bacteria, plant mitogens, PHA, and pokeweed mitogen, and foreign erythrocytes. And we, from a lymphoblast-specific or enriched library of transcripts, we began to see these leucine-rich repeats, an enrichment of them. And a sequence analysis of the first 22 that had a complete open reading frame, or almost complete, we noted to our surprise that each one of them had a different sequence. They differed in the number of these leucine-rich repeat modular units, which they had, and in the ones that they shared, the sequences were also different. This turned out to be the rule in lots of different lampreys, mainly larvae, but adults as well. And out of over 600 sequences, we found very few repeats. This is a stick model of the longest one in the previous slide, and it features an invariant stalk region, but this is the region, this leucine-rich repeat region is the variable part of the molecules. As for many leucine-rich repeat molecules, it has a fairly long N-terminal and C-terminal leucine-rich repeat unit. These are called capping LRRs. They protect the hydrophobic core of these crescent-shaped molecules. And in addition, all of them had an LRR1 of about 18 and connecting peptide of about 13 residues, and in between, variable numbers of these green LRR 24 residue building blocks. There are some interesting features of the stalk region, the invariant part. It's rich in threonines and prolines, perhaps suggesting flexibility, and we noted a GPI cleavage site potentially, and it turned out that expression of a construct of this with a Ha marker by mouse thymocytes indeed brought it to the surface, and you could remove it by cleavage with a phospholipase C of bacterial origin. But there are also about 8 cysteines in this carboxy terminus, and they are important too. When we looked with Chris Amamiya, who had a sperm germline library, we could only find, to begin with, one VLR. We call these variable lymphocyte receptors, or VLRs, because they were so variable and expressed on the surface of the lymphocytes. And it was very incomplete. It encoded for the stalk region and a signal peptide, but only bits of the N-terminal and C-terminal LRR were found. And in addition, large stretches of non-coding intervening sequence. But flanking that incomplete germline gene were coding sequences for LRR sequences that had the missing elements that might, if stitched in, would generate the mature VLR genes, the products of which we were sequencing. And this process turned out to occur only in lymphocytes and not other types of blood cells. So it would appear, I guess I missed, that one doesn't show. So let me just tell you a summary of what has been learned about these variable lymphocyte receptor genes. It turns out that there are 3 of them. They are all incomplete. These are called VLR-A, B, and C. This is the original one, the VLR-B that I just mentioned to you, that could be encoding a diverse repertoire of lymphocyte receptors. And in hagfish, there are only 2 of these that have been found so far. VLR-C is the most recently discovered one by Kasahara and his colleagues in Japan. They code these VLR proteins of the type that I've just mentioned. The way that these genes are assembled isn't known completely, and the biochemistry is lacking, but the donor cassettes that flank these are very numerous. For example, around VLR-B, upstream and downstream, there are over 400 of these donor cassettes. And since some of them are singlets, some are doublets, and occasionally a triplet, there are actually more than 800 potential cassette or LRR unit donors. And that they are brought in here in a random fashion, except there has to be there appears to be a need for a sequence, a short stretch of nucleotide sequence similarity between the donor and the recipient LRR. And so these are brought in, and then the DNA is apparently cut, and the donor sequence in part or pieces is sewed in or is copied in. And that process occurs over and over, along with deletion of these intervening sequences, until one generates a complete mature VLR gene. The donor sequences are not moved. They have no recombination signal sequences, and many of them have stop codons, but they can be used in this fashion to generate a huge diversity. It's estimated by Igor Rogozin, a computational biologist at NCBI at NIH, that the potential repertoire for this process is somewhere between 10 to the 15th and 10 to the 19th potential different types of receptors. Now this process begins on either end. It can begin on the 5' or 3' site, but it almost never occurs on both both alleles. Occasionally, one finds a non-productive or incomplete assembly on the non-productive allele. And so it's only one allele and not the other, begins on one end and not the other. These VLR-A, B, and C can be, cells that express them can be discerned because the invariant stalk regions are all different from each other. And it's possible to make discriminating monoclonal antibodies, mouse monoclonal antibodies, for example, that we've made. So you can distinguish the cells that express each type. And each one of these, VLR-A, B, and C, is expressed by a different lineage of lymphocytes. And the VLR-As and the VLR-Cs appear to be T-like cells, and they are generated, their receptors are assembled. We've learned from studies in collaboration with Thomas Boone that these are assembled in tips of the gill folds, and the primary filaments is where these two are assembled, and that's the only place that you can find the expression of an AID ortholog discovered by Rogozin and colleagues called cytidine deaminase 1. And it's thought to be involved in this assembly process that I've just mentioned by a gene conversion-like mechanism, although not exactly like the ones we know that generate diversity of avian antibodies. Neither of these lymphocyte lineages, they both respond to antigens with proliferation, and they are preferentially, both of them, responsive to stimulation with phytohemagglutinin, or PHA, just as mammalian and other jawed vertebrate T cells are. They don't secrete their receptors. They actually don't bind the native unprocessed antigens that we've tested them against. We know that they are preferentially responsive to alloantigens. So they will proliferate to white blood cells from other lampreys. This is an outbred population. Better than they will to other antigens, and that's not true for this VLRB lineage. They actually look, in many ways, have similarities to the two discrete lineages that we have of T cells, alpha, beta, and gamma, delta, although that analogy has not been carried forward more than that presently. They make, when they are activated, cytokines that the VLRB-like cells have receptors for, and vice versa, suggesting, at least for this A and B lineage, that there may be a cooperative or functional intertwining of these as for our T and B cells. Now, I'm going to spend most of my time on this VLRB lineage, which is more like our B cell lineage. These receptors are assembled, it appears, primarily in hematopoietic tissues. For both of these, if the assembly is non-productive, the cells don't survive outside of the site where they are assembled, our evidence suggests. For the VLRBs, there's an association with the expression of another AID ortholog called cytidine deaminase 2. CDA1 has been shown to have deaminase potential, but so far CDA2 has not. That's currently being studied, but the idea is that it too may be involved in the assembly, but of the VLRB cells. If you immunize, and the larvae we study mostly because they're easier to catch and keep. They only require feeding of yeast every couple of weeks and not fresh fish. If you immunize them by injecting the immunogen into the coelomic cavity, as for example, if you inject human O-positive erythrocytes, within a couple of weeks, all of the animals will make circulating hemagglutinins that agglutinate these human, and these are very specific. If they are immunized again after a couple of weeks, then the titers increase about 20-fold. Now, all of these hemagglutinin, all of this agglutinating activity can be removed by removing with the monoclonal antibody to the invariant stalk region of VLRB. The cells that are expressing VLRB are located primarily in blood vessels, and here you're looking at the gills, filaments, that before and after immunization with anthrax spore, Bacillus anthracis. And the green cells are the ones making VLRB. And after, you see with the BRDU label, these yellow cells indicating a proliferative response that's fairly prominent after immunization with this immunogen. The cells that are secreting the agglutinins, or the circulating VLRB proteins, here is an example of one that looks like a plasmacytoid or plasmacyte, Some look more mature plasma cells. The products that they secrete, is shown in this Western blot with anti-VLRB monoclonal, are very large molecules, and you can see they're off the scale of our standard, and they are variable in size, as you might expect, in that they include different numbers of those green LRR building blocks, and they are disulfide linked. Pardon me. Brant Herron devised a way to make recombinant monoclonal VLR antibodies. The immunization, preparing RNA from the cells of immunized animals and constructing cDNA libraries, and they can be expressed clone by clone in human embryonic kidney cells, and then you can determine in wells which ones secrete the product and which ones are specific. Now, this can be done now much more efficiently by expressing them in yeast surface display method, which allows for high throughput screening and screening for different binding capabilities. This is a cartoon of what they look like. They look much like our IgM molecules. They are made of 5 pairs, sometimes 4 pairs, of identical chains. They differ from our IgM antibodies in that they are made of only one type of chain, and they lack a light chain. They are bound together through these disulfide bonds using those carboxy terminal cysteines that I pointed out earlier, and the last 4 of the 8 are particularly important. Now, the antigen binding sites are these leucine-rich repeat modular units. And using one that a recombinant antibody RBC36 that Brant Herron made, Byung-Woo Hong in Ian Wilson's laboratory was able to determine the structure of this region, LRR, binding this H trisaccharide, which is our blood group O positive determinant. And you can see here by looking at these hydrogen bonds in this ribbon diagram that here's one in this LRRV2, here are 2 more in this N LRRVE, that these are sites. Now, in this carboxy terminal LRR, there is a highly variable insert that I did not mention earlier, and it encodes for a loop, a variable loop that sticks out, and these look much like a curved hand with a thumb at the base. This gray is the H trisaccharide determinant that I just mentioned in the space-filling model, and you can see there's this antigen binding pocket. Now, these VLR antibodies that lampreys make, the ones we've looked at so far, are exquisitely specific for the antigen. This one, this RBC36, for example, on a glycan array of more than 600 different glycans, only binds to certain ones. Others, there's no background essentially whatsoever. And all of them that bind have a fucose with an alpha-1,2 linkage to galactose beta-1,4. Those 2 sugars are essential for this binding. And it doesn't bind anything that doesn't have them. And the Bacillus anthracis antibodies are equally specific. So these are some of the advantageous characteristics of these lamprey antibodies. We call them antibodies because they fit the original description of antibodies being very specific for antigens. They are highly specific. Here's another feature that makes them, gives them some advantage for binding certain epitopes. This VLRB2D was one made by Zev Panzer in his lab, originally with us, and the structure was solved by Roy Mariuzza, whom I believe was at the Pasteur as well at one point. At any rate, they showed that this loop, for this antibody that's specific for hen egg lysozyme, binds down in the cleft or a crevice type of epitope. That's not seen with all of the mouse heavy light chain antibodies. They see curved or planar surface epitopes. It does happen with camelid antibodies and with an unusual kind of heavy chain only antibody made by sharks. They bind with a range of affinity, some with quite high affinity, and I won't show you an example. Even when each one of these binds with low affinity, the avidity is staggering. It's extremely difficult to get them off of an antigen column. Brand had to raise the pH greater than 11 to elute them. Going down to as low as 2 did not elute them from an antigen column. And when he brought the pH back to neutral, they bind perfectly happily again. They're very stable. They survive high temperatures. You can sit them on the bench for months and they still bind particularly well. They're easily engineered because they only have one chain. But this is the point that I'd like to emphasize in trying to, maybe this is not mouse to bedside, but from hopefully lamprey to bedside. We last had a common ancestor probably around 500 million years ago. So it's unlikely that these cells would be subject to the same kinds of tolerance constraints that mouse and human antibodies would be. So we've been immunizing, among other things, with representatives of different stages in our B-cell lineage. And this just shows the different types of B-cell malignancies where the malignant cell type accumulates at each stage along this pathway. And for the one that I would like to show you is one that was made against a human plasmacytoma, fresh plasmacytoma cells, and it's recombinant monoclonal antibody called MM3, made by Gertz Ehrhardt, reacts specifically with plasma cells. And it doesn't make any difference whether it's a malignant plasma cell or a normal plasma cell, but doesn't react with any of the preceding stages in B lineage differentiation, nor with T cells, non-T, non-B cells in the tissues that we've looked so far. Now, this epitope we are particularly interested in because there are no specific target molecules or antigens for myeloma cells or long-lived plasma cells that could be making autoantibodies or making allergic antibodies in severely allergic individuals. So we're concentrating on trying to determine what that antigen is in order to try and find a way to use this discovery platform to reach a therapeutic kind of reagent that could be used in these conditions. So far we know that it's a conserved epitope in non-human primates, so that will give us models to begin looking at the potential of the products that we use. So how did this complex alternative immune system evolve? And in closing, all of you know that this could be an older system than ours. It's Susumu Ono postulated back in the 70s that there were genome-wide duplications, 2 of them. This is a more modern version of phylogeny of chordate lineages, and Amphioxus is at the head of the chordates. The tunicates or sister groups of the vertebrates. And the first round of the genome-wide duplication, the evidence favors occurred before lamprey, and the second one after the jawless and jawed lineages split, although that's not determined with finality, I understand, just yet. So this would have given 2 genes instead of 1, and then 4 instead of 1, a way of possibly hastening some of the developments that led to an adaptive lymphocyte-based immune system that we have. So if this is true, then one could speculate that the type of, although these receptors are totally use different prototypic genes or co-opted genes, and a favorite possibility is this chain in the von Willebrand factor, clotting factor receptor on platelets. This CD42B is only other LRR molecule that's been seen yet that has an insert in this carboxy terminus and has a flexible loop like that, but it's not variable. So it could have been co-opted for, adapted for these systems. It's not possible, of course, to say which is the oldest because there's no way of telling when along these pathways each of these systems evolved. But it's amazing, to me at least, how similar the construction of the lymphoid systems are. And that suggests that these ancestral T and B lineage cells, that bifurcation, and probably for many other types of lymphocytes that we now refer to as innate lymphocytes, that process occurred prior to the development, one could speculate, of these 2 different types of receptor systems. So these are some of the things that we think we've learned, that it's, since every surviving vertebrate has one or the other systems, it suggests they are very important. This construction of interaction may well be essential to, because if you generate repertoires in a random fashion, you're bound to generate self-reactive receptor-bearing cells if they have at least the capability of making pro-inflammatory cytokines, and that's about all we know so far. It's a setup for autoimmune disease. And it never hurts to remind ourselves as we slice and dice the immune system to try to understand exactly how it works, that the whole system evolved over many millions of years to work together. And finally, we hope that they have biomedical potential, at least as diagnostic reagents. And again, my picture doesn't show of my colleagues, but these are people who've been involved in doing this work that I've been mentioning, and many collaborators, many of whom I mentioned along the way, and we've been supported by NIH and by other institutions, and thank you.